Hello, everyone. Thank you for virtually joining us. I'm Pengyuan Li, a PhD student from the Computational Biomedicine and Machine Learning Lab at the University of Delaware. My advisor is Professor Hagit Shakay. Today, I'm glad to talk about our work, utilizing image and caption information for biomedical document classification. Biomedical document classification is an important task for supporting the biocuration process and biomedical research. Most current methods focus on textual contents, which are typically extracted from the title and the abstract. Notably, figures and captions convey important information about biomedical research and the results in publications. Several studies attempt to use image caption information for document classification. Our group is one of the first to use image content information, as both images and captions have shown effective for document classification. In this work, we propose a new classification scheme using both image and caption information. However, there are challenges for using image information. Most current biomedical publications are stored as PDF. As such, it is essential to first extract figures and captions from the PDF files of publications. To do that, we use PDF Capix that was developed by our group for parsing biomedical documents. Another challenge toward utilizing figures is abundance of compound images consisting of multiple types of panels. To use the image information from each individual panel, we use compound image separation system, fixed split, that was developed by our group as well for separating compound images into their constituent panels. Both of our systems are publicly available online for use. As noted, many figures consist of multiple types of panels. To use the image information, we propose a new image representation that encodes the combination of different types of panels in a figure. To do that, we first introduce an image taxonomy comprising 11 categories. We also noted that figure legends or margins are sometimes over-separated from their original image, thus forming individual panels. These are actually separation residues. As such, we augment our taxonomy with a separation residue class. Next, based on this taxonomy, we train an image classifier to categorize the extracted panels. We use an annotated dataset comprising around 35,000 pre-labeled panels. Its statistics are shown in this table. A pre-trained convolutional neural network, VGG16, is used for image classification, and our image classifier attained accuracy of 88%. Once the class label of each panel is obtained, we represent each figure as an 11-dimensional binary vector, C1 to C11, where Ci is 1 if a panel from class I in the figure and 0 otherwise. This figure shows the process for converting figures into their corresponding figure words. For example, figures consisting of histograms and fluorescence microscopy panels are converted to this vector. Its first and sixth value are 1 as histogram belong to class 1 and the fluorescence microscopy belong to class 6. We refer to each of such vector as a figure word I. A document D is then can be represented as a D image by using figure words. In this vector, II is 1 if the ith figure word appears in document D and 0 otherwise. For handling captions, we first concatenate all captions in document D into a single document. We then identify all biological concepts like gene, disease, using pulsator, and applying stemming and stoppwords removal for pre-processing. To generate the caption-based document representation, DCAP, we employ pre-trained word embeddings, which map words to vectors of fixed dimensions. For titles and abstract, we employ similar procedures to generate the title and caption-based document representation, DTA. So far, we have obtained 3 document representations. Next, we employ 2 schemes for integrating the information from all 3 sources. The first integration scheme concatenates vectors of 3 document representations, the image, the cap, and the DTA into a single vector, DO, to represent a document D. We use SVMs, which have demonstrated the best performance in this context and denote the classifier as convey. The second integration method employs a meta-classification scheme. To do that, we first train 3 base classifiers, CImage, CCAP, and CTA, using the representations of images and the captions, and the titles and abstracts respectively. By applying a base classifier to a document D, we obtain the class label L and the probability P to be assigned to the relevant class. Each document D then can be represented as a 6-dimensional vector DO. This representation is then used for training the meta classifier, combine C, which assigns the final class label to each document. We use SVMs as the model for base classifiers and the meta classifier. To evaluate our method, we use 2 datasets. The first dataset, denoted as GXD2000, is a subset of the dataset curated by the Jackson Laboratories Gene Expression Database. It contains 1,000 documents that are relevant to Gene Expression Database and 1,000 irrelevant documents. The second dataset, denoted as DSP, comprises 534 publications relevant to molecular interactions and 448 irrelevant ones. To obtain figures and captions, we first apply PDF capex to these 2 datasets. We next apply fixed split to separate compound images into their constituent panels. The numbers of extracted figures, captions, and panels are shown in this table. To evaluate the document classification performance, we use standard measures: precision, recall, and F-score. In our first experiment, we compare the classification performance obtained when using only a single type of information, whereas the performance when using all 3 types of information. From this table, we can see our classifier, CImage, utilizing the figure words alone, attained F-score of 78% over the GXD2000 dataset, and F-score 73% over the DSP dataset. In order to better understand the contribution of figure words for document classification, we identify the most distinguishing figure words by ranking them using the Z-score test. This table shows top 5 scoring figure words along with their occurrence in the relevant and irrelevant dataset. Over the GXD2000 dataset, we can see there is a large difference between the figure words distribution in relevant dataset and that in irrelevant one. For example, there are 1339 figure words consisting of fluorescence microscopies in relevant data, while only 437 such figure in the irrelevant data. Similarly, over the DSP dataset, there is a clear difference in figure word distribution between the relevant and the irrelevant dataset. Therefore, our figure words have much potential for improving biomedical document classification. We also show examples of top scoring figure words. Notably, these figure words correspond to images that are typically found in certain biomedical experiments. For instance, fluorescence microscopy images are often appear in publications relevant to the gene expression database, as this method is a common imaging technology for visualizing gene expression. Pull-down assays are commonly used in studies relevant to molecular interactions. Thus, figure words correspond to gel images are important for document classification over the DSP dataset. As such, we believe that our figure words convey important information about the biomedical experiments and thus contribute to the document classification task. Next, we look at the performance obtained by classifier CCAP. When captions are used only, and CTA, when titles and abstract are used. It is interesting to find that over the GXD 2000 dataset and the DSP dataset, these two classifiers attain similar performance. However, the relevant publications correctly identified by classifiers are quite different. This figure shows a comparison of relevant publications identified by classifier CImage, CCAP, and CTA. Of the GXE 2000 dataset, 24 publications can be identified by CTA only, while 18 relevant publications can only be identified by CCAP. Notably, 29 relevant publications can only be identified by CImage. Similar patterns can be found from the classification results over the DSP dataset. These findings strongly suggest that figure words, captions, and titles and abstracts provide distinct and complementary information for document classification. Thus, a significant improvement is obtained by combining information from images, captions, and title and abstracts. Our classifier convey attained the highest performance on all measures over the GXD 2000 dataset and over the DSP dataset. In our second experiment, we compare our classification performance to the performance obtained by 3 state-of-the-art systems. Over the GXD 2000 dataset, while the precision attained by our method is slightly lower than that attained by the RFCAP method. Our method convey attained the highest recall and F-score. Notably, recall is often viewed as more important for document classification. Over the DSP dataset, our classifier attained the highest performance on all measures. These results demonstrate the effectiveness of our proposed document classification scheme. To summarize, we introduced a new biomedical document classification scheme using information derived from both images and captions, in addition to the title and abstract. Our experiment and results demonstrate the effectiveness of our newly proposed figure word for representing images and for improving document classification. Our experiments also demonstrate that figure words, captions, and title and abstracts provide distinct and complementary information for document classification. As such, The document classification in performance is improved by combining the information from all three sources. Last, I'd like to thank all my collaborators for their wonderful work and contributions. Thank you very much for your listening. Now I'd like to take your questions.